We have Dr. Rajmohan B, Associate Professor from Government Ayurveda College, Tirunelveli, for a session and the topic on research dimension and emerging scope of Rasashastra and Bhaishajya Kalpana. We wholeheartedly welcome you, sir, for today's session.
Yeah, thank you. So basically, I was discussing with Ramesh sir is there and all other faculties are there. They are all my colleagues as well because I'm also a part of Amrita since the inception of its PG courses. So I'm so happy to associate with Amrita at all time. And again, this is going to be, I don't want to talk much. I just want to make an interaction with the students, what their perception regarding Rasashastra and Bhaishajya Kalpana and what actually they expect and what are the main queries they have in your mind. So we can move forward this as an interactive session to give an initial perspective, initial thoughts to provide a platform for discussion. Basically, we all know that every medical science works in two limbs. They have basically two limbs. The first limb mainly being, advancements in diagnostic techniques. We have our Nidhanasthana and all. We may adopt newer and newer diagnostic techniques so that our accuracy in diagnosis can be improved. Or we can develop our own tools for diagnosis of diseases which are mentioned in our own classical textbooks. And there may be newer and newer diseases emerging as in COVID-19, as in the case of COVID-19, which is caused by a beta coronavirus. And earlier there were SARS outbreaks and of course there are certain newer diseases emerging as the human race is gaining much more age. Naturally, there will be newer and newer diseases. Basically, the diseases are thought to be the contribution of civilization because earlier when the man were forest dwelling, they were subject, and also the lifespan of the human beings were comparatively very less, like 20 or 30 years of age. They lived there, and there were not much knowledge regarding a disease which are happening in the 5th decade or 6th decade or 7th decade. So when the civilization is improving, naturally, the lifespan of the human beings is improving. There will be emergence of newer and newer diseases will emerge. And we need diagnostic techniques and newer diagnostic markers for early detection of these conditions to manage. The entire globe is going after various markers, just like to sharpen our Purva Rupas. Whatever we are getting from the Purva Rupas, they need some markers so that these Purva Rupas can be standardized and which can be utilized for the prediction of prediction of diseases. And whatever the next limb is naturally the pharmaceutical limb, in which whenever there is a new disease, naturally you need a medicine as well, or you need to repurpose the existing medicines to be used for the newer conditions. So the pharmaceutical limb is one of the major area that every medical science should work on. So likewise, Rasashastra and Bhaishajya Kalpana are basically the pharmaceutical limb of Ayurveda. The future of the medicine naturally depends on future of Ayurveda naturally depends on the advancements in drug and device development, and of course both are dependent on Rasashastra and Bhaishajya Kalpana. But as a practitioner of Rasashastra and Bhaishajya Kalpana, I never see such such a rigor in the practice of Rasashastra and Bhaishajya Kalpana, and there is not much utilization of a branch as well. This may be because of the unaware, because of the students or the aspirants from R&D are probably unaware of what they are possessing in their hands. So I think the students need to be more and more aware towards the possibilities of Rasashastra and Bhaishajya Kalpana, and they should be aware of the changing dimensions of the pharmaceutical arena across the globe, and as well as the nutraceutical, cosmeceutical, and phytopharmaceutical industry that are emerging globally. And you should shape yourself to meet the changing needs of the society as well as changing needs of the market. Definitely, health care is also nowadays turned to a market-based service. So that it's controlled by insurance giants, it's controlled by marketing giants, and it's also done by some of the big pharmaceutical companies as well. So, we should change, or we should approach our R&D in such a manner that every R&D student should have that ability to be fit into the newer dimensions which are possible with nutraceutical, cosmeceuticals, and all those things. These are the main thoughts, quick thoughts I just want to introduce here. So you can debate with me, you can discuss with me, and you can also voice your queries, which will definitely going to, we will have a wonderful dialogue, so that will be beneficial for all of us. That's it from my side.
So, sir, recently I just came across an article which gave emphasis on reverse pharmacology on most of
Okay.
in Chandraprabhavati. And it got me thinking that we already have so many preclinical and the clinical studies which prove that prove our the effect of our medicine, and how is that we can further do these analysis to make our science to establish it as a true one.
Okay, so doctor, you are telling that there are sufficient enough studies are available to prove the effectiveness of Ayurvedic drugs. That was your first statement.
Effective
Yes.
are there, but how can we explain like how this medicine works in various pathways?
Yeah. Okay. Yeah. Unfortunately, I want to, I'm not in a position to agree with your first statement, because there are not sufficient studies exist to 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 prove the effectiveness of our medicines in certain dimensions of healthcare. Okay, there is a we know how our medicines work, and all the Ayurvedic drugs which are explained in the classical textbooks are taken for granted for its effectiveness. That is the way we do it, because the World Health Organization is telling that you can directly go for a phase 3 trial for an Ayurvedic drug if it is mentioned in the Schedule 18 drugs, which are mentioned, 18 textbooks which are mentioned in the Schedule 1A of the Drugs and Cosmetic Act. That means all these medicines have proven efficacy, and it is written in scriptures. It is there in your textbooks, which can be taken as a proof of its effectiveness after observing after having a long-term observation for centuries. So that can be taken as an effectiveness for is a proof of effectiveness. But public health, the term public health means medicine. The term medicine means that is an interaction between a doctor and a patient. If you prescribe a medicine to a patient, we call it as the application of medicine. But whenever some mass disease occurrence are happening, and you are announcing that this particular drug is very useful for masses, that has to be followed by an evidence-based approach, like means like that of a proper research methods that should accompany. So public health language is something different from what we are using in our classical textbooks and all. Now, we never use Vadrekta in terms of public health, but it may have a different sense. We may not be able to use Jara. When you have to, whenever you are telling that a particular drug is useful in COVID-19, a particular drug is used in post-COVID therapy, you have to prove the effectiveness of that drug which you are using, which you are claiming. If you are claiming it for post-COVID therapy, naturally you have to prove its effectiveness in using it in public health, because that drug is going to be to the mass in the public health. But as a doctor, when you are prescribing it to the patient, there is no conflict, because you can prescribe any medicine to a patient by following the principles of your medical science. But if you are aiming at a global health or a public health, the thumb rule is that there should be an evidence-based method that we have to follow. For that, definitely we need much more approaches. One of the thing is that the standardization of the products. Now I will just tell you what are the processes that requires qualification of a drug to be used in a public health. Okay. So the first thing is that the drug should be standardized. Whatever you are purchasing from Thiruvananthapuram, that the same quality and quantity of the active ingredient as far as an Ayurvedic drug is concerned, the plant part you are using, or the mixture you are using, or the avaleha you are using can be considered as an active ingredient. So the concentration of the active ingredient should be, that should be standard across all the places, or wherever you are getting the drug should be standard. So to ensure that standard, the raw material should be standardized. So the quality is the first step that you have to assure that. So whatever when X company X is, Amrita is having, Amrita Life is a pharmaceutical company. They are producing Agastya Rasayanam, and Kottakkal is producing Agastya Rasayanam. Naturally, these two Agastya Rasayanams, the profile will not match. Maybe it will not match. It should not be like that. Agastya Rasayanam, if your marketing is an Ayurvedic medicine, it should have a property, it should have a desired quality across any companies, or across any places, or whatever it is. So that is going to be one of the challenges that we are facing. So ensuring a uniform quality across batches, or across GMP certified companies or thing. You take paracetamol. As far as paracetamol is a single molecule, and the standard concentration of paracetamol in a tablet, as well as the profile of paracetamol in a tablet, chemo profile of paracetamol in a tablet, is all remains the same. If you take it from any companies, or if you take it from from across any place you collect, that remains the same. So that exists a huge difficulty for us to standardize our product. So that is the quality researches. The researches relating to quality of Ayurvedic products, that is one of the areas that we need to follow. The next thing is that the quality. Then comes the safety and effectiveness. Safety first and effectiveness second. Safety means how much safe are these drugs while we are administering to human beings. Of course, we have a lot of documentation. All our textbooks are telling that these drugs are safe, and World Health Organization is actually telling that, of course, if that is the case, you can take the safety is taken for granted. So the toxicity studies or something, that may not be necessary for Ayurvedic drugs. But if you are not going to ensure the quality, naturally the safety aspects, you also need to conduct the safety, go through the safety studies and all those things. Next comes is that the dose of the product. So the safety comes from various aspects. If you really want to have a new combination of Ayurvedic drugs, you are trying to create a patented product, a patented proprietary product of Ayurveda. Naturally, the safety also is one of the considerations. You are preparing a specific phytopharmaceutical from an Ayurvedic combination. You are developing a newer drug from Ayurvedic products and all. Naturally, you need to have this safety profile as well. Safety profile comes from, there are in vitro safety studies, in vivo safety studies, and bio. There are various other mechanisms to look into the safety of various drugs. Then comes in vivo, again animal model, and then you can move further to phase 1 trials, phase 0 and phase 1 trials, and address all these issues. But that is regarding the safety of a drug. If it is a classical drug, you need probably that safety part you can reserve, because it is already known to everybody that, and it is also written there that it is safe. So that safety study is followed by what is the dose of the drug. So that is the major area we are challenging area as far as R&D persons are facing. We are telling that the general dose of Avaleha is 1 pala or Krida is 1 pala. And for certain prescriptions, maybe Ramesh sir is prescribing 1 tablespoon. Maybe I am prescribing 1.5 tablespoon. What is the base of this prescription? Do you feel that we have a standardized dose for our formulations? What is actually the dose of Kalyana Ghridam which shows effectiveness? So, these kinds of the posology study, I think that also need to be a very critical area that Ayurvedic, Rasashastra and Bhaishajya persons need to address. That is, we don't have a standardization of dose across for the same formulations across prescriptions. So that is another thing. So the calculation of dose as per the modern pharmacology is appear to be extremely difficult process. There are two processes are there: pharmacodynamics and pharmacokinetics. Pharmacokinetic studies means absorption, ADME, absorption, distribution, all those things you have to metabolism and elimination studies, and also the bioavailability studies. How much percentage of active ingredient are going right into the bloodstream or right into the serum? Of course, we have polyherbal formulations, and of course we can identify a specific active constituent from that polyherbal formulation. Then we focus on that active constituent, how much percentage it is coming into the blood. Based on that pharmacokinetic studies, basically you need to fix the dose and duration and the dosing of the drug. How much amount you have to take, how much quantity of drug you have to take, and after the repetitive dose, after how many hours you have to take the repetitive dose. So this is a very tough study. That pharmacokinetic and pharmacodynamic studies that encompasses a very larger area. Perfusion studies are there, cell membrane crossing studies, cellular uptake studies, a whole lot of studies are there. Unfortunately, I have never seen such papers are coming out with in these areas, which are very critical as well. That calculate, even if it is a single herb, we are not conducting these kinds of pharmacokinetic and pharmacodynamic studies, which appears to be very critical in calculating the actual dose of a formulation, along with the toxicity studies. So that is the safety area. Then comes the effectiveness. If a drug you are using for the classical indication, as explained in your textbook, there is not for the clinical studies are necessary. But if you are repurposing it for another disease, because you are utilizing, Agastya Rasayanam for CASA or Agastya Rasayanam for Shwasa conditions for centuries, that's okay. But if you want to repurpose that for post-COVID related lung fibrosis, you need to prove the effectiveness. Safety is established, but you need to prove the effectiveness in that particular condition. So that necessitates at least a phase 3 clinical trial to be used in a public health. You can prescribe that for your personal patients. That's not an issue. The doctor is always having a freedom to practice medicine. That's fine. But when it comes to the public health, if you are announcing to the public health that telling every doctor that you should prescribe this medicine for lung fibrosis, you have to follow this procedure of evidence-based integration of products and practices to public health. So naturally, that is the thing that usually we have to do. Again, you have to go for the effectiveness studies, efficacy studies, that is phase 2 study, and then go for effectiveness studies. And somehow, you also need to go for the pragmatic trials, that are phase 4 trials. After the drug is widely available in the community, you collect the ADR, whatever the adverse drug reactions which are reported from the drug, the administration of the drug from the medical practitioners. Now it is, the patients can also report ADR issues arising while they are taking medicines. So I think that Amrita is an ADR cell, and Amrita is doing wonderfully in the field of ADR as well. I think the R&B is controlling that. Again, the ADR cell is controlled by R&B. So that is the way in which these things are being done. So reverse pharmacology is, reverse pharmacology. What is the advantage of reverse pharmacology? Means, whenever you are, amlaki is having many properties. Okay, so which angle you should study amlaki? That is provided by the Ayurvedic description, therapeutic description of amlaki. Amlaki is Vaisasthavana Sveshta. So it may have regenerative potential. So we already know the pharmacological activity of that plant. Then we go back and look into, what are the mechanism of action of that drugs, why it is having regenerative potential. I also studied amla as a part of my PhD studies, and I have found out that amla is acting like resveratrol. Resveratrol is an anti-aging drug which is found in the seed coat of grapes. That's why red wine seems to be appear to be anti-aging. So the same manner, there is a sirtuin-1 gene protein is an a sort of metabolic pathway, is acting on metabolic pathway, and it can act as an anti-aging, provide you with anti-aging properties and all. So basically, the same pathway amla is working and act as an anti-aging, anti-aging potential. Now we are studying we have one of our project which we are looking into two rasayanas, which Shodamshravi rasayana and Amalakyavi rasayana for its regenerative potential in zebrafish models. So likewise, we already know that this is the pharmacological activity of this particular drug. And from that activity, we are moving back and we are observing, why those activities, like which protein or which pathway is responsible for this. And somehow, now we can have these metabolomics and proteomic studies and transcriptomic studies in which, the entire system or entire, what are the changes occurring in a cell as a whole when you are administering a medicine to a cell, that we can study nowadays. We have the instrumentation, we have the methods available for such studies. So that's why that's how it's being done. I think I have answered your question. During the process of explaining, did I miss what you have asked? Are you satisfied? I'm not. No, I'm not. You're mute.
I'm satisfied, sir.
Okay. So that's it.
Thank you.
Good afternoon, sir.
Yeah, good afternoon. Go ahead.
I am a first year PG student joining 2025 batch.
Okay.
So, as you were talking about Rasayana, I had a question like, can you throw some lights on limitations we can face if we bring Rasayana as a nutraceutical application? Traditional Rasayana.
So first part of your, I didn't hear your first part of your question. What was that? Rasayana only I heard.
No, I like you to throw some light on if we take Rasayana as a nutraceutical preparations. If we try to bring Rasayana as a nutraceutical preparation, then what can be the limitations we face? What we might face if we go for such an experiment.
Yeah, rasayanas, as you are aware that rasayanas are of two types, Vadadipika rasayanas or Kudi Praveshika rasayanas. The rasayana drugs are very peculiar, Rasayana drugs have, if a particular drug to act as rasayana, specifically I'm talking about Kudi Praveshika rasayana, you have to follow the rasayana vithi. That is important. Rasayana vithi is important than the rasayana drug itself. So there is a procedure mentioned in our science which makes Chavanaprasha a Kuti Praveshika Rasayana, because if you take Vadadipika Rasayana and Kudi Praveshika Rasayana, Kudi Praveshika Rasayana is always Sreshta than Vadadipika Rasayana. So all the potent Rasayana drugs you have to take it as Kudi Praveshana. So the Kudi Praveshana Rasayana Vithi is making Chavanaprasha as a more potent Rasayana drug. So you can take Rasayana, you can take all the Vadadipika Rasayana, all the Kudi Praveshika Rasayana as Vadadipika Rasayana as well. But the problem is that Vadadipika Rasayana will not have that much of effectiveness as that mentioned for Kudi Praveshika Rasayana. So these are the two demarcation between Rasayana, Kudi Praveshika Rasayana and Vadadipika Rasayana. Vadadipika Rasayana means, you don't need to follow the Rasayana Vithi. But in Kudi Praveshika Rasayana, you have to follow the Rasayana Vithi, strictly follow the Rasayana Vithi. Rasayana Vithi is making the Rasayana more effective. That is important. So if you want to have the full effect of Chavanaprasha Rasayana, you have to administer that in Kuti Praveshika. The Kuti, how to prepare the Kuti, how to take a Panchakarma before entering into the Kuti, and which are the dietary restrictions you have to follow, which are the behavioral restrictions you have to follow, and the lifestyle modifications you have to adopt while you are administering this Rasayana Vithi. So that is known as Rasayana Vithi. Okay. In our classical textbooks, there are several mentioning regarding this Rasayana Vithi. Natam atyubhimji the Rasayana Vithim vina. It is explained for Leshuna Rasayana. Natam atyubhimji. You should not use Leshuna Rasayana Vithim vina. You can use above the dose of, I think, 4 pala is the dose. So you can use Leshuna Surasa more than 4 pala only with Rasayana Vithi. or you should stick on to the classical dose of Leshuna. Otherwise, if you want to break that, you only use it as per the Rasayana Vidhi. Majority of the drugs, its usual dose is lesser when compared to its Rasayana Vidhi. Rasayana, when it administered as Rasayana, there are many drugs. Kanmadam is there. So these are the difference between a Rasayana in a Kudipravishika Rasayana and Vadadipika Rasayana. We can only convert a Vadadipika Rasayana and a dose as a nutraceutical. Nutraceutical and Rasayana that usually do not match. We provide Rasayana. Generally, we all have a feeling that Rasayanas are regenerative and rejuvenative, and they are Brahmana. It's not like that. Not all Rasayana drugs are Brahmana or, Vadapitta Shamana, or not like that. You take Palladaka Rasayanam. Palladaka Rasayanam is a Kudipravishika Rasayanam. Is that Brahmana? No, it is a Bhalladagani, Tishnani, Pakinya, Samanija. It is as equivalent to Agni. It is not a Mathurya like thing. But again, you go for Tuvaraga Rasayana. There are certain Rasayanas like, that Bala Rasayana. Bala Rasayanam is basically, Brahmanam and all those things. So Rasayanas are Labhopayo Sastanam, Rasadina and Rasayanam that can improve the Agni Deepanatva, that can improve the Sodoshotanatva, and all those things, which is the basic swabhava of Rasayana. And indirectly, that can also, along with the food and dietary restrictions or whatever you're taking, that can act as regenerative and regenerative body. So that is the basic thing. So not all Rasayanas can be converted into nutraceuticals because, there are Tuvaraga Rasayana, Bhalladaga Rasayana, etc. are there. Nutraceutical means, you select certain drugs and you look into the properties and you can say Tila Rasayana. Tila Rasayana can be, I have felt several times that Tila Rasayana can be converted as a nutraceutical and you can administer in the perimenopausal age. That would be very helpful because in that age, there are diminished, ovarian hormones can cause, loss of calcium as well as, the psychological factors are also playing there. So you can have a sort of Pravalabhasma combined with Tila Rasayana can act as a very good nutraceutical at that age. So that depends upon the activities of the drug and it should be a Vadadipika Rasayana better. And you choose wisely based on its indications. Then you can convert any Vadadipika Rasayana into nutraceutical. It is explained like that, the majority of the Vadadipika Rasayanas are explained as to improve endurance or improve promotional activity, health promotional activity. You can select such Vadadipika Rasayanas as definitely as nutraceuticals by observing its activities and skillfully placing it in a target population, just like what you are placing Tirasayanam in perimenopausal, as I suggested. This is it. Hope you have got the answer, right?
NIC, finally a PhD scholar. As a part of one of our project, we have done the cellular ion uptake of Shodhita Kasasa with the two titration mediums, Pringaraja and Nimbusarasa, and raw Kasasa, Shodhita Kasasa also. And the result was after in vitro digestion, we have done the cellular ion uptake. Maximum ion uptake was seen in Nimbusarasa. And next, it was seen in Rokasasa. The least was seen in Pringarajasarasa. Pringarajasarasa, Shodhita Kasasa. So we were a little confused on what may be the reason for maximum was seen in Nimbusarasa, Shodhita Kasasa. Almost double ion uptake was there. But second was Ashodhita Kasasa. And very least less amount, almost 0.125% only for Bringarajashodhita Kasasa.
So you got a comparatively better cellular uptake for Ashodhita Kasasa than Bringarajashodhita Kasasa, right? Yeah, yeah. We need not worry about these findings because you have done it in the method is that you have done the digestion and then you have administered in the cell line, right? That was the process. Okay. So maybe one of the reasons maybe the Bhringaraja Shodhita Kasisa may be using for a particular condition, and the other Shodhita Kasisa may be using Nimbusarasa or Shodhita Kasisa may be using for another purpose. That may be the reason why. So whenever we are doing these studies, we already have something in our mind that you are all going to administer it. You perceive that in the sense of the management of anemia, right? You want to be have a specific amount of iron in the blood, just like they are telling that for us humanity is having this much of percentage of iron delivery and all those things. You think you make it think it in a different perspective. Likewise, you are having an external application of Kasisa, like a Kasisa the labum, and all those things are there. You are directly applying on the affected area in which that penetration we should look for. So in that sense, maybe there you maybe Prangaraj showed the Kasisa can be used for external application or something like that. So it depends upon the mode of application as well as the condition which yeah, which you are administering Kasisa. The uptake, the percentage of uptake comes into play. That is one of the things. Again, while we are doing the experiments and all, naturally, there may be many bias, and also there may be certain factors that will influence the findings of the study. Probably the phytoconstituents which you are making Pungaraja, there may be something, the particle size after triturating may be affected with the some of the particles which are present in the some of the phytoconstituents which are present in the Bhringaraja, or that may be help to clog the particles. So that this clogging may be, once the particles are clogged or flocculated naturally, the cellular uptake will be reduced, even if the particle size is the same across all the formula you have used for cells. Some, if the fineness, that depends upon the fineness and clogging of the particles. For certain materials, this clogging may have formed clumps. These clumps may not be not that much easily absorbed through these cells. So likewise, I'm just providing you one of the possibilities, which what might have happened that may hinder the absorption of that particular type of Kasisavasma you have used. So I think all these factors should be considered while we are possibilities we should be considered while we are before we are discussing or concluding what we have obtained in our studies. It may not be the reason. Typically, may not be the reason of it is the property of Pringaraja that is hindering it. It may be a physical property that is promoting the clogging of these phosphors that may be hindering the absorption, uptake from the cell. From the phrase, I can read that you have still some confusion existing there. You may please ask a follow-up question to make it more clear.
Sir,
or I can make it in a very simple. I can make it in a very simple statement, which is very simple and straight. Unless and until we go much deeper into this process, and we need to control the experiment in various stages, we cannot say that whether there is an actual reduction in absorption of Pringaraja Sorasam, Pringaraja Sorasa Bhavita Kasisa Pasma, or is there any external factors that is affecting the absorption of this particular Pringaraja Bhavita Kasisa Pasma. We have to control these stages of experiment, and we have to do much detailed study, which is out of the scope of a PG study. What you have observed here, you can report it as such, and you can suggest that this doesn't mean that Pringaraja Bhavita Kasisa Basma will be absorbed from the gut in a slower manner, or a less uptake from the gut will occur if you administer it orally. We cannot conclude it like that. But no, there is what we are observing here is this one. It may have many factors, you experimental factors that might have affected the. That all those factors also need to be identified and controlled prior to make such comments, I think.
Sir, with respect to the nicest question itself, if you want to write article, can we keep the Brangaraja thing, take only two things, Ashodita and Shodita, and say that iron absorption will be better with doing Bhavana in Limbu Suvasa?
Sir, sorry, sir. There was a call came to me and I missed in between. But what was the question, sir?
So, if you want to make it as an article, actually these are not the thesis also, it's a project given for the second year. Because as we do have
okay.
auto-digestion and cellular uptake study in nature itself, in the second year,
Okay, sir.
this is a project. So now with this,
Okay, sir.
writing an article is difficult. So if you write an article, leaving the Brangaraja part, taking only Ashodita.
No, sir. No, sir. Because that is not a fair mean to report an experiment. My suggestion is that if possible, you need to report it, and you also need to identify some of the factors which might have affected the absorption. All those possibilities that exactly will alert other scientists as well. These kind of a finding is already there, in which Brangaraja Shodhita, iron is less absorbed in the process. So I think that is the right way to report that. And if you found that there is go through the methodology again. If you found that there is anything happened during the process, or there is a methodological flaw occurred only for this Brangaraja, you can eliminate that and you can report. But if you are not able to identify any methodological flaws with respect to this Brangaraja, causes of asthma, I think you should report it, because if any other person are also doing the same method, replicating the same, they also will get the same result. So I think if you are confident with the methodology, my suggestion is to report this.
The only thing we missed, of course, it was out of our budget and time also. The estimation
exactly.
Of quantitative assessment of iron after the auto-digestion, if you would have done, maybe in Brangaraja been less, because Brangaraja generally compared to Limbu, Brangaraja will have more phytochemicals. Might have increased the quantity, resulted in the reduction of iron, so that it would add. And of course, if it is a pure Ayurveda concept, I think we can interpret like Brangaraja is indicated in Kamala and Limbu is indicated in Pandu, because Kasisa is indicated in Pandu. Maybe Brangaraja iron uptake is less, maybe acting at the liver level.
Exactly. And again, one of the factors which we may consider is the final pH of the product. Whenever you are mixing, processing a phasma, and what was the final pH in solution of this particular thing. Whenever you are, whatever solutions sometimes you may DMS or something prior to giving to the cell line. What was the pH, exact pH? pH is one of the factors that can strongly affect the cellular uptake. So that is a simple thing which we can do. You take the Brangaraja Shodhita, Kasisa Vasma, and all other things, and Ashodhita Kasisa Vasma. You dissolve in whatever medium you are about to give to the cell line. can we just simply check the pH, whether there is any pH variation is there. So that itself can affect the absorption, uptake.
Myself, Dr. Manjula.
Okay.
Sir, for chronic diseases, we have to use medicines for prolonged use. So how come Rasashastra can work in this chronic diseases? Can we use for a longer duration?
Yeah, that's a very good question. And basically, you should know the toxicity profile of various resolutions. That is very important. Whatever medicines which we are using for in any system of medicine for chronic conditions, like you just take the rheumatoid arthritis. It's a very methotrexate is one of the drug of choice. Do you believe that methotrexate is a safe drug? It's not. It's basically, it's a bone marrow depressor. That will cause in long term. It can harm your body like anything. And whenever you go for an eczema condition or a psoriatic lesion, the basic drug of choice maybe sometimes it is external and internal application of steroids. Do you believe that a steroid therapy is very safe? Never it's safe. Something like that. Whenever the disease is severe, naturally the choice of drug also should be like we take a higher order drug. When you take a higher order drug, as far as Ayurveda is concerned, it is Dhatu Viparita. Basically, whatever we are starting, your initial treatment is Ketu Viparita. Next is Dhatu Viparita. Why a Dhatu Viparita drug we are using? Dhatu Viparita drugs are meant to take away, meant to pull out the Dhatu Leena avastha of a disease. Basically, it is there, the dosha avastha is closely interacting to the Dhatu. Something is very closely interacted with the Dhatu. If you are removing it from the Dhatu, naturally that Dhatu will get damaged. That is the part of the treatment. Okay. So, you cannot avoid it in the treatment. That's why there are clear indications, clear directions telling that, in Garbhini and in children, you should not use Dhatu Viparita Chikitsa. Why? Because, that will naturally interact with the Dhatus and that may affect the growth and development of the children or growth and development of the fetus. So that's why, that direction is there, very clear direction. It's a generic direction, but it is very crystal clear that, where and when you have to use these formulations. So that is the first thing. So which condition you are aiming at? You are aiming at a cancer. So naturally, the disease itself is very devastating. And, whenever you're administering a cancer medicine, sometimes, the person die with not because of the cancer, but because of the medicine they have given. The same when it comes to Ayurveda, naturally that creates a lot of issues. Even if it is a cancer patient, you are administering it, suddenly there is some occurred, the people will suddenly start telling, or even you will be very doubtful regarding the drug you have taken. Whenever you are taking a medicine, the majority of the drugs are passing, majority, not all the medicines are passing through the liver. There is a orally taken that will be a hepatic bypass effect will be there. That means the majority of the medicines will have its primary metabolism that is happening in the liver. These chemicals are not accustomed to, the liver is not accustomed to these kinds of chemicals. That means there will be loss of liver cells with the majority of the applications of majority of the medicines, including Ayurvedic drugs or, whatever modern pharmaceutical drugs are there. But since we are all aware that since the liver is having the capacity to regenerate or, to cope up with all these damages, that will be fine. That whatever damage happened, that will be reversible. We call it as reversible damage. But the one thing that you have to keep in your mind that there are very specific directions regarding the use of Rasashastra which are mentioned in our science. You take a disease, you make sure that you want to use Rasaganthimagara, you want to use Talagabasmam, or there are the toxicity profiles of drugs are there. Rasashastra, Rasasindura, or mercury-containing formulations are comparatively safe. And when you go through the literature, existing literature in regarding the toxicity of Ayurvedic drugs in PubMed or Scopus or whatever it is, you may not find that much of case reports or something that describing the toxicity that is occurred from Rasashastra. That means mercury-containing formulations. The major culprit is being lead and arsenic. So I would suggest that lead-containing formulations, you must be very careful. Arsenic-containing formulations, you must be careful. Then it comes to Valsanava-containing formulations, you must be careful because Valsanava is toxic and allergic to certain people. Then you have to focus on mercury-containing formulations. The mercury toxicity may occur not in liver or something. Mercury is mainly eliminated through, large part of mercury will be eliminated through the kidney, so that there may be kidney damage may be much more prevalent than what we usually expect from neurological or hepatic damage. So, but the problem with the lead is that there is not much problem with the mercury, and there exists considerable problem with the lead. Because mercury, the absorption of mercury or mercuric sulfate from the intestine is only 0.01%. That itself is enough to create a therapeutic response. So but the toxic dose is somewhat very high. So it's quite very tough for a person to take Rasashastra even for 1 month with 125 mg 2 times a day. The absorption is comparatively very less. It's very tough to get such a thing. But when it comes to mercurous chloride or mercury chloride, its solubility is much high. And the dose as per Ayurveda is also very, very comparatively very less. It is Desakarpura, you have to mix it 1 or 2 grains, mix it with water, and you take 1 or 2 drops of that water. That is the dose. How effectively Rasatarangani has standardized that dose. So that also, based on that absorption, it's comparatively less. But the problem with the lead is that lead in the inorganic form, mercury in the inorganic form is very, very, very less absorbed from the intestine. But lead in the inorganic form, that is lead sulfide, 100% bioavailability is there. So, lead will cause so much of issues. So if you look into the literature, you can see that all those studies are basically, they contain Mahayogaraja Gugulu, which contains lead. And again, arsenic containing formulations, again, arsenic containing arsenous trisulfide or disulfide or arsenic trioxide, that is Gauri Pashanam, is highly so it's soluble in water, and its bioavailability is very high. That's why 5 milligram dose is enough. That's why it is known as Akupashanam. Akupashanam means it is a toxin. Likewise, it is already there in the textbook, but we forget our these basic things. So the thumb rule is that, basically, a drug which can be give continuously is, let it be like for me, it is 14 days. Beyond 14 days, if you are not getting any response from a patient, what is the point of continuing that drug? Basically, in Ashtanga Sangram, it is only 7 days. Saptahena guna alabhe kriya manyam prayojet. Saptahena, 7 days. Guna labhe, you are not getting the what is intended. Kriya manyam prayojet, you select something else. So, you will get a response. For every treatment, this depends upon disease by disease. For a viral fever, 7 days is, 7 days itself, viral fever will vanish. So, 2 or 3 days, you must get a response. If you are not getting a response, it's a natural regression of the disease. It's not because of your treatment. Naturally, the disease is regressing. But for rheumatoid arthritis-like conditions or Vadashondita is a broad umbrella in which many disease comes. Basic many disease come. A response of 2 weeks time, that is naturally you should expect there's a reduction in pain. If you are not getting that, do not continue the medicine. And if you want to continue your medicine beyond 21 days, give a break. Give a break, allow the body to eliminate whatever it has accumulated, because all these metallic the Rasashastra have a tendency to accumulate in the body, and it can produce cumulative toxicity. So, that you can give some time, so that will be eliminated, and give an accurate dose as per the weight and as per the, the basic metabolic rate. That all those factors should be, the dose should be standard, the duration should be standard. So these are the thumb rules that you need to adopt. And the pathya, apatyas, sometimes you have to take the food, sometimes you have to take it in empty stomach, and the associated things. Certain drinks you take that will facilitate the absorption. Certain drinks you take that will, basically, there are many examples I can provide. Mercury containing formulations may be then absorption may be enhanced with the presence of proteins or something like that. And again, malathion, parathion, like organophosphorus toxins. If you are having egg white with you or protein with you, that will be largely absorbed from the gut. So the absorption of iron is hindered with the presence of milk or calcium products, and vice versa. So likewise, you have to wisely advise, arrange your pathyapatyas and all those things, and it is so important. So my suggestion is that continuous use of Rasashastra beyond 14 days should be justified, both in the toxicity, in the safety level, as well as in the effectiveness level. That's it. A maximum I prefer in 14 days, or 7 days is majority of the medicines. If you are not getting a response, I will drop. If I'm getting a response in 14 days, then we will put a break. And then for some medicines, 21 days. So which one, the drugs which you can safely you can use is Suthavarga drugs, which are very safe. You can use Amprada very safely. You can use, it's a collection of minerals, collection of micronutrients you can use for a long time, pretty long time without all much. Of course, Gandhaga containing medicines. Again, you can somehow it's a without any other minerals you can use. But again, the problem with you have to be very careful with what should I say, Valsnabi containing. Sometimes it may, like, the problem may not be the chailia which is associated, maybe the Valsnabi which is associated. That's it.
Good afternoon, sir. Myself,
Good afternoon.
Dr. Anjali, First Year PG Scholar. Sir, can you suggest some areas where we can start a research topic? From which all fields can be taken for our thesis, like that?
Some areas. Yeah. Actually, it's a shortcut. But basically, I never do that, because the problem is, as far as a first year student is concerned, it is rather than to do a research, rather than to find a result, uh, you should learn the process of research. That is more important than your finding. So there are certain steps which we have to follow while you are while you are delving into your research. The first thing is to identification of a research problem. Okay, from where you are getting research problem, you discuss with your colleagues, you discuss with your teachers, you discuss with your seniors, you teaching is the best method to identify research problems, because some of our ideas, what we obtain while we are taking a class, suddenly one studious student may stand up and ask a question, probably we may not have an answer. So to find that answer, probably we may go for a research. So likewise, that is the research process. As I said, Rasashastra and Kalpana is a huge area. There is a wonderful area for a lot of research. I would suggest, we can have a drug development area, device development area in our institution. Probably at least a couple of students will every year. Nowadays, we are focusing on a device development. So that is one of the area. For drug development, there are many areas: nutraceuticals, cosmeceuticals, and area. For the drug to be developed, quality is one of the issues, and safety issues, effectiveness issues. So these are all areas I'm just telling you. And market studies, how the market responses, what are the issues that is really happening in the industry. Have you ever gone to an industry and observed, what are the problems faced by the challenges faced by the industry? If you really want to understand the challenges taken by the industry, you have to, interview the industry itself. So all those things, come into play while you are about to discuss a topic of choice of Rasashastra and Bhaishajya. And you can do in vitro studies, in vivo studies. There are many, 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 many areas. If you keep on talking like us, you will be more and more confused rather than more and more, gaining insight into which subject you need to take. I would always suggest, what are your, major, interest? What do you really want to become after doing an R&D? Okay, there should be a question there. You should answer this question. Yes, sir, I want to be a clinician. Then you do something which is relating to, of course, if you want to become a clinic, which area you want to focus. So you select a topic that based on your that area. So I want to be a researcher. Then you focus on which area you want to research. Then you select a topic which can nourish your idea of research and your area of research. So likewise, I want to be an entrepreneur. So you try focusing on some drug and device development or something like that. So it depends upon your area of interest and depends upon what do you want to become after completing your R&D. So take your time and think about answer these simple questions, and then you select a choice of your topic and discuss with your guide. Whenever the students always have a thinking that this is my topic, it's not like that. It's the topic of the department and the topic of the guide along with the student. So you should have a consensus with your student and the department before you taking your subject, because the guide should support you as well as the department should provide you the necessary facilities so that you can easily carry out the research. But whenever a student is telling research, Ramesh sir is having plenty of years of experience. He would easily predict, whenever you are taking a subject, he may have a 360-degree idea of the student because he has seen so much of students. So they will suddenly get whether you are able to, your life conditions, your background, they know that you will be able to complete this work or not. That the teacher may be knowing, but suddenly he is refusing you. You may feel very awkward. Why sir is not allowing me to do that? Probably he may not be, or the teacher may not explain you or every aspect. But there may be some reason why they are telling. So you politely ask them, sir, why I cannot do that. So usually every teacher will explain to you why it is so. So you take a consensus, then you carry out the research. But I would probably, to begin with, I would suggest you to go and read the R&D theses which are already conducted in your department, and you constantly read some papers relating to pharmaceutics and which are the areas, pioneering areas which are happening in the general pharmaceutical areas, shelf life studies, everything is so much of important. And what are the industries need? What are the current industry, Ayurvedic industry really in needs? So you look into all these aspects and you weigh against your personal choices. Then you would probably come up with a very good research idea. Okay.